A very good afternoon, all the participants. I believe post-lunch session is always difficult to handle, right? But I'm pretty confident that our keynote speakers will really make the session very fruitful and as well interesting. So there is a small deviation in the schedule what we have given because of the health issues. Dr. Kenami Kilik is unable to join us today. So we have made slight changes in the schedule. So we'll be having two keynote speakers today's afternoon session. So now let me take the pleasure of introducing our first keynote speaker of this afternoon session, Dr. Lakshmi Parida. Dr. Lakshmi Parida is an ISCD fellow, IBM fellow, master inventor, and leads computational genomics at the IBM Research Center. She is a member of Mathematical Sciences Council of IBM Academy of Technology, ISCB International Society of Computational Biology, Association for Computing Machinery, Society for Industrial and Applied Mathematics, and is a visiting professor at the Courant Institute of Mathematical Sciences, New York. Her research spans cancer genomics, neurological genomics, plant genomics, population genomics, and topological data analysis. She has published over 200 peer-reviewed research papers, edited 8 volumes, and authored a monograph on pattern discovery in bioinformatics. She holds over 50 US patents. She has received numerous awards in recognition of her work at IBM Research and external awards such as the Cacao Innovator Award. She is on the advisory board of New York Engineering School and editorial board of BMC Bioinformatics, Journal of Computational Biology, and associate editor of IEEE ACM Transactions on Computational Biology and Bioinformatics. And because of, as she is currently in US and because of the time mismatch with our conference timing, she could not come on live, but she has shared her video, which we can share right at this point of time. So, sir, can you play?
Greetings from New York. I will talk about topology in Big Dataomics. I see data in two flavors. Either they exist in a coordinate space, which you may know as a point cloud, or it is in a coordinate free space or a relationship space. Of course, the number of points that you have in the point cloud is no longer a problem. It is the dimension of the data that leads to difficulties in this space. So does the dimension affect even the coordinate free space? But first, let me take an example from the coordinate space and apply statistical learning to solve a problem. And we have been in the cancer genomics space for a while now, and we have an active project with broad on understanding drug resistance. We have been working in an offering that IBM has called Watson for Genomics and so on. So we have been in this space for a while. And the next problem I choose is from this space of cancer genomics. So the question that we ask is, does the signal of a disease subtype exist in the genome? Okay, and we are looking at hematological cancer. And why are we looking at subtype of a disease? Because the types of disease, the signal for types of disease, the gross types of disease is perhaps not as complex as for subtypes. And the problem is complicated because the lesion may originate in the same hematopoietic stem cells. In this case, this is 7 different kinds of blood cancer we are looking at: AML, CLL, CMML, and so on and so forth. So we apply a statistical method, so AI technique more or less agnostically, and try to separate these disease subtypes. And here there are about 700 patients. Every dot corresponds to a patient, and the different colors are the different subtypes. As you can see that, and we have applied some algorithm on separating it, and you can see that separation is not very good because it has formed one big cloud there. And we try with different parameters, as you know, with AI methods and so on. There are many hyperparameters that you try, and we separate a little bit, but not so good yet. So to cut a long story short, we had to go back to the drawing board. We developed our own AI algorithm and introduced what is called stochastic regularization, and used tensor learning and applied this. And we used we designed these methods based on what we were learning about the data. And so lo and behold, after this, we find that the data separates very cleanly. Of course, this is a TSNE plot of the data, but it gives you a sense of the fact that the algorithm is separating the data, or the signals exist in the genome. So the next question is, where on the genome do these signals lie, right? We're looking at the whole genome. So we divvy up the genome, and we look at whole genome, and then we split it into genic regions and non-genic regions. The genic regions is split further into intronic regions and exonic regions, and the non-genic region, we split it into annotated parts, that is, those parts of the genome which scientists think is functionally useful, or functionally has some functional implications. And you'll see that all these regions that I talked about, that they lie in all the chromosomes. So the two concentric circles that you're seeing on the outside correspond to the different chromosomes, going all the way from chromosome 1 all the way to chromosome 22 there. And now that I've explained the infographic, and these different regions correspond to different parts of the genome, like I mentioned here, now we can see how well they perform in separating the disease, or how much of signal do they carry in separating the disease. And this, to make it simple, I'm showing in terms of color. The darker the green, the better the separation. Lighter the color, the worse the separation. And here, when we use off-the-shelf AI on the original data, you can see it's somewhere, it's sort of mediocre. It's just about, it's doing medium well. It's not doing terribly well. And when you permute the data, when the data is permuted, it is light, it should not separate, and it is not separating as expected. But when we use a new method, you find that the disks have gotten darker. So the separation has gotten better. That means the algorithm is working well, and the permutation data has even gotten lighter, suggesting that perhaps something is right here. And you may think that because these areas covered on the genome are not the same, maybe the area is playing a role in separating these subtypes. So we do another sanity check by normalizing this area, so taking them as equal lengths, and you find that they still separate, and these different regions still separate. What is not clear from the figure, because I'm showing these to you by colors, is that the region which is called the dark matter, the one that we don't think has absolutely any functional implication, is the one that does the best separation, indicating that dark matter matters. That is, the region that we thought has no functional implication, has no implication, actually is doing the best in separating, suggesting that there is a functional link there. Again, the skeptic could think that, perhaps these methods, since these are agnostic AI methods, are they actually taking some noise? Are they mistaking some noise for signals? And so we tried this on some new neurodegenerative diseases that we have been studying, this ADNI and Michael J. Fox data, Parkinson's data, genomic data, and actually it does not work. It does not separate as expected. So if it had separated here, one would have been a little worried because one would think that it is perhaps getting some noise as signals. So to summarize, this suggests that the dark matter, what we think as a dark matter, perhaps has some implications, and perhaps scientists should be spending some time and energy on looking at this region, and we need to understand these regions. We got this as our results, and there have been other researchers also seeing similar results, coming to similar conclusions. So this was an example of using AI methods on genomic data, and the dimension here was very high. The dimension say here is in terms of SNPs, whereas the number of samples were reasonable, about 1,000. And let me put in a plug for an upcoming book on rethinking cancer, which is addressing the dark matter and other types of such discoveries or such new things that we are finding out about cancer. Then moving on to the next topic, which is the coordinate free space. Just as you have the dimension, the curse of dimensionality in coordinate space, such a similar thing would exist even here. And the tool that I'm going to be using here is topological data analysis, or TDA. And the plot I'm showing you here is somewhat in the spirit of what we understand as the curse of dimensionality for the coordinate space data. And this, and I'm kind of plotting the difficulty, which is the y-axis, the difficulty along the and the x-axis represents the dimensions in the coordinate free space. So are you looking at pairwise information? Are you looking at three-way information, four-way information, and so on? And the difficulty here is the number of simplices that we get, or number of edges, hyper edges and edges, and you can see that it grows exponentially with that. So this also has a similar problem. That does not mean that we cannot use it, just like we have been using statistical learning. And I'm going to circle back just in case the people in the audience are not familiar with TDA. I will give a short primer on TDA, assuming that people are not familiar, so that all of us are on the same page with this. So let me begin with a simple example. So here is, say we have a graph with 11 vertices, and B0 is the 0th Betti number, which is keeping track of number of connected components. What we see on the left is the same as what you see on the right. Now let's add a few more edges. So we add 7 edges to this. And you know the Euler characteristic, which is the alternating sum of vertices and edges, 11 minus 7 is 4. And you see that B0 is 4 number of connected components that you see. Let's add a few more edges. So we added 2 more edges. Now the alternating sum is 2. So what happened? Because we have introduced now 2 cycles, and hence these 2 cycles are captured by B1, the first Betti number, which is 2, B0 is 4, B1 is 2, and the alternating sum now you can see in the 2 columns is 2 in both cases, 11 minus 9 and 4 minus 2. Now, moving the space of dimension, I want to introduce a hyperedge between 3 vertices, or what is called a 2-simplex, and I introduced that as the triangle that is shown here. And the cycle that was represented in time t3 by this is now becomes a boundary of this, and it is no longer a hole, no longer a cycle, and instead a few more cycles are introduced by the other 2 diagonal edges that I've added in the rectangle below. And if you are seeing more than 3 3 cycles, you're actually seeing correct. Yet B1 is 3 because the number of independent cycles is still 3. I'll come back to this point because the number is 3, but you can identify different sets of 3 cycles as independent cycles. So there is no unique way of saying which are the cycles from this B1 number equal to 3. And then to make it a little more complex, I add 4 more 2 simplices to this. And now we introduce a hole that is in the tetrahedron there, and that is now beta B2, or the second Betty number, is equal to 1. So we were actually tracking these cycles or these holes at different dimensions, and this is captured by what I call bar diagram. So you can see B0 is going all the way from T1 to T5. So it began with 11, and then from time the second time onwards, it is 4 number of bars, that is 4, that is retained. And B1 you can see at time T3 there are 2 bars there, and then one of them dies, and there are 3 bars between T4 and T5, and so on. And B2 at T5 there is one that is born, and so on. So this actually captures the birth and death of bars or homologies. Okay, homology groups. And the template that I will use throughout the talk would be as follows. We'll see what are the input, what is a simplicial complex. There are different kinds of simplicial complexes like Beatrice, Rips, Check, and so on. So type of simplicial complex, and what is a filtration? How is a filtration defined? So here in this example that I used, the simple example, the filtration was that we had the first simplicial complex, which is fully contained in the second, fully contained in the third. That is, we are only adding more and more simplices, but we are not removing any simplices. And we are going from left to right by adding more and more simplices. And this is called and this is defined as a filtration. And the second point is on additional structure. I will talk a little bit about this in the next slide. And the additional structure, as I had pointed out before, is the fact, for example, at time T4, we saw that B1 is 3, but it does not uniquely map to 3 cycles. And so sometimes if you want to interpret your output, you'll have to go from what you have in your output, map the output back to the input, and you might want some structures that allows you to do that. And the output is in terms of bar diagrams. I'll show you different ways of using the output. So we define this notion of essential simplices. This gives us, this is a technical definition, but it gives us a way of mapping your output to your input. That is, the output are these bars or the homologies. It gives us a unique way of going from these homologies to your input. Essential simplices simply means that these are the simplices that are present in all the representations of the homologies or of the bars that you see. So I will quickly give you 5 different ways of using these bar diagrams. We have used them in different contexts. I will go through these examples to show how these bar diagrams can be used in different ways. There can be no bar, which is called a mapper, or you can use all the bars and get a signature. You can have multidimensional bars, or you can use individual bars. You can have formulas around the individual bars, or you can prune and interpret each individual bar. So let me begin with an example of using all the bars as a signature. For this, I choose the problem of admixture and from population genomics. What is being shown here are an evolving population. Each circle here is an individual. Each row is a generation. And at the bottommost row represents the current generation. The top row represents the previous generation. And there is an edge between these nodes. indicating the transmission of genetic material from one generation to another. And these are humans, so each node is going to have two parents, exactly two parents, one mother and one father. So it is so its genetic material is actually a mixture of the genetic material of its mom and dad. And this one you'll realize is going to have closed paths, not cycles, because if you consider these edges as directed, they will have closed paths. This is not a tree. So it is essential to recognize that this is not a tree because of recombinations or because of mixing of genetic material from two parents. Now, if I take a 60,000 feet view and move back, then I begin to see populations emerging. For example, you can see in the central structure here, maybe you had a population which split into two different populations. They evolved independently. And then you can see somewhere down in the middle, there is a mixing of two populations and so on. So you have four populations, A, B, C, D, and the populations A and D are not admixed. The populations B and C in the center are admixed, which is clear from this figure, clear from this bird's eye view or the 60,000 feet view. But when you have genetic data, you have data only of the bottommost row, and you have only the genetic markers of this, and that's all. You don't have this history. So how do you solve this problem? So we actually proved a theorem which says that the highly persistent homology of the Vietoris-Rips complex of the structure, the arc structure or the graph structure that you see on the rightmost, is approximated by the persistent homology of the Vietoris-Rips complex of the scaffold, which is the structure on the left. Okay, and these two actually can lead to using a distance matrix, because you can have a distance matrix which is defined on the leaf nodes or on the bottom sample of or the current population. And this suggests that there are large, highly persistent homologies, large cycles and small cycles, and there is some chance of separating the small and large cycles. And we use this in the context of plant genomics. We use them on avocado. This was work with USDA, and the motivation for the problem was to find the diversity that existed in avocado, to see which were admixed and so on. So we use this algorithm, and what we are using here is the signature of the bar diagrams that you can see. So admixed and non-admixed cultivars, when looked at together, give you different signatures. So to summarize, the simplicial complex here is a Vietoris-Rips complex on the samples, and you are doing a filtration of the simplicial complexes using genomic distance, and you don't need any additional structures here, and the output is, of course, the bar signatures, bar diagram signatures. The next example I'll use is on a formulaic use of individual bars, and this I choose from the space of metagenomics. Again, we have been working on metagenomics problems for a while. We have worked on the sequencing the food supply chain problem, and we are also now working on the healthy aging project, AI and healthy aging with UCSD. So we have been in this space for a while, and this problem I choose from this space. So here is the problem, which is the problem of signal enrichment of metagenome next generation sequencing reads. So what is the problem here? Is that you are looking at the reads from an environment, from an entire environment. So it is not a single organisms, but you're looking at multiple organisms together. So you're getting the short reads of all these multiple organisms together. And what is the challenge and what is the problem here is that the reads can be small. As a result, they map onto multiple reference sequences. And And this is a database, say, of microorganisms, and these microorganisms are very similar to each other. So, and these reads would map onto many of these reference sequences. Now, the question here, of course, is you want to actually separate the false positives. What are the false positives from the true positives that you are seeing? Now I want to take the same problem and try to use less information, because we use a sort of a complex math tool of TDA. Can I use less information? Okay. And so this is an experiment where we take the same problem, where I don't look at the genomic coverage and everything like we did, we just did. Instead, I just look at only the number of reads in this overlapping region. So now it is a simpler problem. Now I crack this through my TDA treatment again, okay? And so we do the filtration. So we use the essential simplices. Again, we use a scoring function, which is similar to the scoring function that we had used before. But of course, this would be different, slightly different, because we have actually changed the problem for this. Now I will cheat a little bit and show you some of our best results. And the results I'm showing you here is the ordered list of these organisms. And what is shown in green is the known true positives. This was not simulated data, but this was a controlled experiment where we knew what the true positives were. And what you are seeing in blue is the result of a method which was not using TDA, which was using the classical method. So it looks like TDA got it right, and the classical method, at least in this example, did not work. So I showed you that we got more from less, in the sense that I used less information, that is only the number of reads, and nothing more like length of the reads or what is the fraction that they cover, yet I was able to solve the problem. And in this case, it outperformed a classical method. I'm not saying that it will universally outperforms, but at least here is an example where it outperformed a classical method. So to summarize, the simplicial complex here again was a check complex on reads, and the filtration on the simplicial complexes was based on the overlaps between these sets, and the additional structure was the essential simplices, and the output was the formulaic use of bars. Now I'll show you an example where we'll be using the mapper, that is no bars or 0 bars, and we will also be pruning and interpreting these individual bars that we have. Of course, we are in the midst of a pandemic. The reason why I'm not in Moscow in person, which I regret, but the pandemic is raging, and more than almost 1.5 million deaths already, more than 50 million infections. And early on this year, when the pandemic began, IBM also responded to COVID-19 by making many resources available to the research community, like the supercomputing grid, the IBM Genomics platform, and so on, at no obligation, at no cost for researchers and even for users. And we also started working in this area of COVID-19. So we started looking at problems from this space. As many of you know, that the first genome was released in early part of January, and after about a month or so, the disease was named as COVID-19 and the genome was named as SARS-CoV-2. And people started looking at the different genomes that were looking at the genetic differences between the viruses that were being released. We were also amongst them. So around summer, July or so, we were looking at about 60,000 genomes, because they're not all identical. And we were doing the classical way of looking at it is through phylogeny. But you're already looking at about 60,000 units now, and the phylogeny structure begins to get more and more complex. And about a month earlier, you will see that the number of genomes have even doubled. The number of genomes that are publicly available for analysis have doubled to more than 120 to 130,000 genomes. And now if we want to visualize, now this is for scientists, if you want a tool, if you want to visualize all the genomes together simultaneously, the phylogenetic tree, if not impossible, becomes more and more difficult to use, right? So we are using a mapper here, where this is an alternative way of visualizing the genomes, this large number of genomes. Again, each ball here is capturing each little node here. This is an interactive plot. So the second figure is a zoom in version of the first one, the bottom part of the first figure that you're seeing, and each ball actually corresponds to a collection of genomes, and they belong to a ball because they're genetically similar, genomically similar, and the different colors refers to the different regions from where they were gathered. This has been the work of our summer student, Sarah, who worked with us, again, remotely, virtually, and she was very effective even while being virtual. There's another way, another problem we looked at based on the genomes of this virus, but asking a different question. And the question here was to study the geographic spread of the disease, not based on infections, but based on the genome of SARS-CoV-2. And what are the kind of questions it answers is that you see that at some time period, say in early part of February, we were seeing that there were two different paths of infection, say from Pennsylvania, two different routes of spread from Pennsylvania to Washington, one via New York, the other via Ohio. And this cycle kind of dies around February 20th. And what does it mean? It means that after that, one is not able to distinguish, it disperses, and you do not see any distinct path of spread. And similarly, you see another red cycle, say around end of February, which showing two alternative routes from Washington to New York, one via California, one via Louisiana. By the end of March, this is not visible anymore. And how does this help, or what kind of questions it helps us answer? You can do a similar exercise. Now, this was based on states in the United States. You can do them on different countries and so on. And this probably helps us understand or ask questions retrospectively about, say, superspreading events or the relationship between SARS-CoV-2 and superspreading or any other related information as well. And so to summarize, the simplicial complex that we were using was a Vietoris RIPS. And here, each node was a state or a geographic point that you'd say in the left, the different states in the US, and you had the genomic information about the virus. You had other metadata, meta information like when they were collected, the dates of collection, and so on. And we use this date for filtration, and that is how the simplicial complexes were. The filtration was defined. It does not require any additional structure. And the output, of course, the first one, the mapper, was a no-bar case. And in this example, for example, this one that I'm showing you here of the geographic spread is based on individual input. And I would also like to go to another interesting example, and that is of using multidimensional bars. And for this, I go back to the example that I had showed you earlier when we were looking at the geographic spread of the disease and the simplicial complex. So when you're talking about multidimensional, here we are in 2 dimensions. So you see a matrix on your right. So if you focus only on the bottom row, okay. So the bottom row bottom row is again a filtration of this simplicial complexes as you move from left to right, and each simplicial complex is contained in the next, and so on. And this x-axis is the filtration by dates, and that was an analysis that I had showed you on the earlier part of the talk. But now we have the y-axis here, and this is in terms of this is haplogroups, or so what are haplogroups? Haplogroups are groups of, in this case, viruses, which have a specific patterns of mutations, specific signature patterns. And why are certain groups of SNPs identified as haplogroups? Because these are implicated in the phylogeny or how they have evolved. All right. And so in this 2D matrix that you see here, if you can you can define your filtration as any track, as long as you're having your simplicial, as long as you're having the containment of the simplicial complexes as you move along, right? And for example, here let me show you an example where in this multidimensional persistence, what I'm showing you in dimension 1 and 0 is that each row, you can see it changing. Each row, the bars corresponding to each row is shown with time. So with the animation here, or the time that you're seeing, is the rows as you go up the matrix. Okay, so this is one way of visualizing it. And there could be many. Like I said, you could have pulled out any other filtrations as well. And the ones that we chose systematically was these along each row, the filtration along each row, and then we are studying it systematically. And in this application, so what is the interpretation of this? Again, you could have a different application, and you can have different interpretation. So the interpretation here that we are using is that we are studying the geographic spread not from individuals who have gotten it, but from the signatures in the viruses that are sampled from these patients. So they're already subsampled, because not every patient is being, every patient's genome is being sampled, the SARS-CoV-2 that is. And so there is a subsampling there. And we are only looking at variations in the genome of this sample. So how do we know that this is robust? And this, in fact, using the second dimension of haplogroups, because haplogroup is yet another way of capturing similarity between viruses, because along the first axis, when we are using near or almost the same viral signatures in the viruses that we see. Haplogroup is a slightly different way of looking at the similarities, and we are looking for robustness of this. So, and as you would have seen in this figure in the animation, it did not change too much as you went from row to the next row to the next row to the next row, and so on. So that indicates that this is fairly robust, and we can build our conclusions quite confidently from this. And in fact, we applied this to the same data set that I had showed you before. So again, this is a quick animation that you can see here, and you can see that it is stable. The structure is more or less the same. And what you notice is that in all of these rows that you're seeing in this multidimensional persistent homology that we did, the cycles, the length of the cycle increases. You have this long cycle at around day 60. So when you have a long cycle and it persists, you can interpret it. A rough interpretation of this is that it is not dispersing as much. when it disperses, it becomes rampant, and you cannot actually detect this anymore, and the cycle vanishes. The cycle begins to vanish when you cannot actually see any distinct paths of geographic spread. But the long cycle indicates that distinct paths remain and it is not dispersing as much. And what is very interesting is that we took another data set, another mode of data set, which is completely different from this, and that is a data of NPIs, or non-pharmaceutical interventions, from this paper that is shown at the bottom here. And this lists the different NPIs that were introduced in the different states, and non-NPI interventions would be closures of school, or closures of public transport, closures of, say, flights, or flights between states, and so on. And this is a data set that simply collects these kind of interventions, and they also peak around this day 60, suggesting that the genomic data and the NPIs are probably telling us the same story. And finally, before I conclude, I want to show an example where we use a combination of coordinate space and coordinate free space method, that is statistical learning and TDA. The example here is Parkinson's disease, which is, now this is not genomic, but it's transcriptomic data or expression data, and this is case control. And we have about a total of 400 and about 260 of case and about 160 of control data. And we are using both machine learning and the TDA. So the data is split into training and test. So for the training data set, we generate a gene skeleton, and that uses a distance which is sort of based on correlations on these training samples. And so here is a cartoon of what this would look like. And then next, these skeletons are individualized for each of these subjects. And then you extract a signature out of this. And this is and here we are using a Vietoris-Rips complex. So assume that each data point that you have, there's a little ball that is growing, and that is growing at the rate of the value of that expression in that gene of that subject. And as these balls are growing simultaneously, the rate is determined or defined by the expression value, they begin to collide with one another. And so you have an intersection of these balls. And so you can now use Vietoris-Rips complex that is tracking the intersection, like I had shown you in many of the samples before. And you can extract these signatures, and now you learn on these signatures. And it turns out that when you use TDA and CNN together and machine learning, it does better than just doing machine learning alone. In fact, 3 out of the 4 measures, it outperforms the different machine learning methods, suggesting that these two can be combined to improve results. So we are working on a system called Matilda, which is a multi-purpose toolkit for TDA. It can be used as a library, or it can be used as a set of standalone applications. And I had mentioned that there is an issue of explosion in terms of size here, and so we are devising new ways of handling this explosion by using approximations, and we are designing new algorithms for that. We are trying to do efficient computation of persistent homology on that, and this supports different kinds of inputs, different kinds of outputs, and if there are any special structures like I had showed you before. And once this is under construction, we are still building it, and once it's ready, we will share with the community. And before I close, I want to show you one of the pipelines here that we've actually used on some real data set, which we have used on Alzheimer's data, where we can use there's a complex matrix of methods, like we can use machine learning, we can use pattern discovery, like the redescriptions, we can use persistent homology, and we can use some domain-specific algorithms and so on in different ways, so that, and to generate hypotheses. So we can mix and match and combine this to answer the complex questions that that we are facing. So to summarize, I showed you an application of AI in dark matter DNA, and presented 5 different applications of TDA, each using a different mode of the output. The mapper, NOBAR, for the SARS-CoV-2 data, for visualizing SARS-CoV-2 data, the signature, all bars for the admixture problem, and multidimensional bars for the geographic spread of SARS-CoV-2. And for individual bars, I showed you formulaic use in the space of metagenomics for signal enrichment problem, and individual bar use as pruning and interpreting in the COVID-19 case. And finally, I showed you that combining AI and TDA, as we did in Parkinson's, can sometimes give you better results than using any one of the methods alone, so that these methods can work in synergy. So the work that I presented today is a work over many years, many people, many collaborators, and you can find more about this work and other work by either going to the link shown at the bottom or simply scanning the QR code if you can. And I'm very grateful to all my collaborators, to their entire teams, and to my team for their diligence and dedication, without which none of the work would have been possible. And this is a picture of my team. Of course, this was in pre-COVID-19 times, and we hope that we will soon be back in a post-COVID-19 phase. And I'll stop here. Thank you for your attention.
Thank you so much, madam, for enlightening us on AI and applications of topological data analysis. Now, moving ahead to the last keynote speaker of today's afternoon session, it is indeed a great pleasure to introduce our second keynote speaker of today's afternoon session, João Manuela R.S. Tavares, to the august audience. João Manuela Tavares graduated in mechanical engineering from the University of Porto, Portugal in 1992. He received the MSc and PhD degrees in electrical and computer engineering from University of Porto in 1995 and 2001 respectively, and the habilitation degree in mechanical engineering in 2015. He is currently a senior researcher with INEGI and an associate professor with the Department of Mechanical Engineering, Faculdade de Engenharia, University of Porto. He is the co-editor of more than 40 books and co-author for more than 35 book chapters and 600 articles in international and national journals and conferences. He holds 3 international patents and 2 national patents to his credit. He has been a committee member for several international and national journals and conferences. He is the co-founder and co-editor of the book series Lecture Notes in Computational Vision and Biomechanics in Spincher. He has been a co-supervisor for several MSc and PhD theses and also supervisor for several postdoctoral projects. He has participated in many scientific projects as a researcher and as a scientific coordinator. His main research interests include computational vision, medical imaging, computational mechanics, scientific visualization, human-computer interaction, and new product development. So now, may I request Joa Manula to kindly address the participants, please?
Okay, good afternoon. So thank you very much for the introduction. So it's a great pleasure to be here, and I would like to thank the organizers for the invitation and to give me the possibility to share with you the work that we have developed in the last years. So let me share my screen. Okay. I think that you are seeing my PowerPoint, yes?
Yes, sir. Yes.
Okay, good. Thank you very much. So welcome to my talk. The title is Image Segmentation by Computational Techniques. Dot line you can see here. So a brief introduction to the topic. Then I will talk about techniques and applications related to image segmentation. Finally, the conclusions. And then I will just show you the names of my students that have been working with me. And then I will talk about some publications and events that we have organized in the last month. So this is my CV. I was already introduced. So I'm a professor at the Faculty of Engineering of the University of Porto in Portugal. This is my school. Just to give you an idea about our school, this is the Faculty of Engineering of the University of Porto. Here we have more than 6,000 undergraduate and master students, almost 900 PhD students. We are more or less 600 technical staff. And of course, nowadays, the situation is different because of the COVID situation, but we have more than 900 international students, usually. Okay, this is the campus of the Faculty of Engineering, all the buildings where we have the classes, labs, and so on. We are more or less 50,000 people working here. So Porto is the second biggest city of Portugal. The capital is Lisbon. We are in the north part of the country. Portugal is this small rectangle that you can see here. So we made border with Spain and of course with the Atlantic Ocean. This is a picture of the campus. And well, our city is known because of the Porto River and because of the port wine. So port wine is a brand of the city. This is one of our more recent buildings that we have in the city. Is the Music City Hall of Porto City. Okay, so let's start with the scientific part. So this field of image processing analysis is becoming more and more important, even now with the COVID situation. We can find algorithms of image processing in several areas of our society. For example, in natural science, sports, biology, industry, engineering, but of course related with medicine for medical exams, diagnosis, and so on. Okay, so for the people that are not so related to this area, what is an image? Well, I have an object of interest, then I have a source of energy. There are different kinds of source of energy. It can be external to the object, it can be internal, but I will have energy that is emitted or reflected by the object of interest. Then I will have a sensor that will acquire this energy, and then I will have a sensor that will build an image. And if it is a 2D image, as I have here, by the way, this image is very known in the field of image process analysis, is Lena. Lena was a playmate in the last 60 years. And since the 17 years, it's used for benchmarking and so on of algorithms of image processing. But anyway, this is an image, a 2D image in grayscale. And an image is like a matrix. So we have columns, rows, and if you zoom a particular area of the image, if you zoom in, then we can see the basic element that is the cell of the matrix. Typically is a square shape, is a square pixel. And the pixel has a number associated. For example, in a grayscale image, you have a number value between 0 and 255. If it is a color image, then we can have, for example, 3 numbers for the 3 color channels, red, green, and blue. But of course, it depends on the color system. Anyway, it's a matrix. So applying mathematical operations, and that can be combined with artificial intelligence techniques, like, for example, machine learning, you can extract high-level information from your input image. For example, if I have this basic image here, I have a square in black, and then I have a white background. If I want to extract the borders of this rectangle, if I apply this mathematical operator, that is a matrix, I will apply here. So that means that the new image, the pixels values will be calculated using each of the pixel values and possible the neighborhoods. Multiply by these weights. So if I apply this mathematical operator, then I will obtain the vertical lines of the rectangle. If I apply the transpose, I will get the horizontal lines. If I sum these two images, I get the border of my rectangle. Of course, this was made in the Cartesian space. I can go, for example, to the Fourier space or other model space. But this is the idea. I have a matrix with values. I will apply mathematical operators that can be combined, as I told you already, with artificial intelligence techniques, and then I can extract high-level information from the image. But anyway, even for us that we have a very powerful visual system, even for us, for example, this image can be difficult to analyze because you have a lot of noise. Even the text that you have in this image, this is a biomedical imaging of ultrasound imaging system. Even for us, can be very difficult to know, for example, what is the region of interest. And here is this area here that is from the carotid artery. Even if I ask you to pick a particular car can be challenging because you have a lot of cars, so you have a lot of occlusion and so on, a lot of objects and a lot of occlusion. Here, this is also a biomedical engineering. You can see that even the imaging processing device can insert some kind of noise. For example, these bubbles here that are from reflections of the light system of the device. And here you can see that part of the image is occluded. So if it is occluded, I don't have so much information from this area. So it will be always very difficult to analyze this area here. And this is a well-known kind of image that sometimes you can find in magazines and so on. For example, some of you can see here a old lady, but others can see a face of a man. So the analysis of image, even for us with our very powerful system, can be very challenging. So if it is for us, will be even more difficult for computational machines. Okay, this is a pipeline of image process analysis. So I have image as input. Then the first step is try to enhance the data that I have in the image. This is known as imaging preprocessing. For example, I can try to remove noise. I can try to correct some geometrical distractions. Then I try to extract features. Or can be the region of an object. It can be just the border. It can be lines. It can be points. It can be just features that then I can use, for example, for 3D vision to extract 3D information from an image or a set of image. I can track the motion. I can try to establish correspondence between objects or between image. I can try to align the image, for example, for data fusion. If I have an exam from CT and an exam from MRI, and if we are from the same patient, from the same organ, then I can try to align the two data sets so I can obtain a more realistic model for that patient. I can do motion analysis, I can do classification. So this first step is typically known as image preprocessing. I try to enhance the data. And when I go to these steps here, is known as image analysis or computational vision. So the idea is to extract high-level information from the image, the data that is already preprocessed. Okay, so starting with the topic of image segmentation, the goal is to identify the object of interest or at least figures of object of interest. There are several approaches for this. Of course, The simplest one is based on thresholding, but then you can go to get algorithms that are more complex. For example, region growing, template matching, statistical, geometrical, physical modeling, and of course machine learning. And nowadays, more and more deep learning. So if you develop an algorithm for image segmentation, sooner or later you need to see if the results are good or not, and you can also compare your results. And for that validation, delineation made by experts are very important. So you can use these manually then made segmentations to use as grand truths, and then you compare the computational results, the computational segmentations with the manual segmentations. But you can also compare your results with the results obtained by state-of-the-art algorithms. And for both of these comparisons, of course, you need to have common data sets. And I have here an example. Here it's a web page where you can find several data sets for biomedical imaging analysis, where you can find the grand truths and the data to segment. So you can apply your algorithm to the data set. You can then use the grand truth to compare, and then you can see if your algorithm is the best one or not, because it's possible to have a benchmarking of the results. And for this comparison, you need common data sets, okay? But you also need common accuracy measures to have a metrics of the quality of the results. There are several metrics. Some of them are based on region information, like Jaccard measure and the dice coefficient, and others are made based on the distance, contour-based polyline distance, for example, or these distances are also very common to compare results. Starting with techniques, this is the simplest one. So it's a thresholding, it's known as thresholding. The idea is I go pixel by pixel of my input image, then I compare the pixel values against a threshold value. If it is above that threshold level, I consider that the pixel belongs to the object of interest. If not, I will consider that it's part of the image background. And here you see an example. I have here a several set of images that were obtained by an optical device that is represented here. Is made of several layers, and if the system is well calibrated, when you have a person walking on the layers, we'll obtain an image. And if the system is well calibrated, the intensity, the value of the pixels is related to the pressure applied. So this is this is very important for analysis of the pressure distribution along the foot sole, and this is particularly important to study the interaction between the foot sole and the ground, mainly for people with diabetics disease. So you have the input image acquired by using this device, then you need to extract the object of interest, and we apply the threshold technique to obtain the region of interest in each of the image. Okay, but sometimes you don't know the threshold value, so it's difficult because this works well if the object is well separated. You have two classes in your input image, otherwise we'll fail. And another approach is known as region growing. So here the idea is, well, I will know for sure a seed point, a point that I know that belongs for sure of the object of interest, and I try to analyze the neighborhood. If the pixels of neighborhood have a similar property, I will consider that they belong to the object of interest. So the seed grows, and then I do the same approach to the new neighbors, and so on and so on. So that's why it's known as region growing. Here we apply this also simple approach to segment this structure here in a CT image. This is a CT image of the ear, and the goal was to segment this bone that we have. This is the smallest bone that we have in our body, is known as bone labyrinth. So the expert, so it's manually initialize this process. The user selects this pixel as a seed, and then the process automatically finds the other pixels that belongs to the object of interest. Of course, this works well if the pixels of the neighborhood have a similar property, like for example, contrast, gray level, and so on. Sometimes this doesn't happen. But sometimes in my application, I know the geometrical shape of my object of interest. If I know the geometrical shape of object of interest, then I can build a template for that object. For example, here you can see a template for the human eye. You know that the human eye, we have the iris, so the iris can be represented by a circle. Then we have the upper and lower arc of the eye. So I can build this template. This template is parameterized, so we have parameters here that change the values of the parameters. I'm able to change the position of the template. I can also change the size, and then I can also change the rotation. So this template can be adapted to a new image that I want to segment that is somewhere there is an object that has a shape more or less similar to the template that I have here. So whenever I have an image to segment, I locate the template to the image, and then I try to deform and adapt the template to the object that is in the image. And for this adaptation, this process is driven by forces. So I need to compute force. I need to establish the force that will driven the template to my object of interest. And to obtain these force fields, I apply mathematical operators based on the features that I know that my object has. For example, if I want to segment the human eye, I can try. I have here an input image. I can try to find the regions that have black pixels, dark pixels, as I have here. But also the whitest regions can be also important. But also the gradient. So I apply the gradient operators, and then I find the borders of the object that are in the image. So using this information, I can establish force that then driven my template to my object of interest. Here we are only segment the iris, so we are using just the circle of the template. It starts here, and then it goes. It finds the center. And then you can see that it starts with a big radius, and then the radius is adapted to get the iris. And here in this image, it's possible to see all the template working. First of all, we find the iris using this approach, and then when the iris is established, we start the process of the matching of the upper and lower bottom arcs. And you can see here at the end that the template is completely adapted to the human eye. Okay, so this approach works very well. It's very robust whenever your object of interest has a shape that is maintained in the other image that you want to segment. Is more or less the same. But sometimes this doesn't occur. So another possibility is to using statistical modeling. For that, you need to have a training data set. So this is a difference relatively to the others approach. You can see here a training data set that is made of 9 images. And so the idea is, okay, I have a training data set that is representative of my object of interest. I go to each of the image. I segment, typically, manually, I segment the object of interest. This segmentation is made of a contour. The contour has points. So if I have points, I can know the coordinates of the points. I can align all of the points of all the contours that I have here in each of the training image. And then I can apply to the all data sets, to the all points, coordinates of the points. I can apply a well-known technique that is the principal component analysis. And with this principal components analysis, I will obtain the mean shape of my object of interest that was learned from the training data set, but I will also obtain the modes of variation. So whenever I have a new image to segment it, that of course was not part of the training data set, I can use this mean shape for a first segmentation, and then I will have the mean model that will be adapted to the final configuration of my object of interest, driven again by forces that are calculated using intensity information, gray level information. But now the deformation is not a free deformation. The deformation is controlled by the modes of variation. So this is a very robust segmentation approach. Whenever your object of interest, first of all, you need to have a training data set, and your object of interest has a configuration that is well represented in the training data set. Otherwise, if the shape is very different, this process will not segment completely, successfully the problem, because we don't have mode of variation that allows that deformation. So here is an example. I have an MRI image. The idea here of the head, the idea here was to segment the vocal tract for speech production studies. So the vocal tract is where the air flows during the speech. And to segment this area, we use this approach. We compute the mean model. So here is the mean model. And after some interactions, you can see that is more or less well adapted to the final shape of your object. Another use of this technique now to segment the tongue during also speech production. An example and another example. And at this last image, you can see two contours, one in blue and another one more or less red. And the idea was, okay, I have in blue the segmentation obtained by the computational method, and in red the segmentation delineated by an expert. And so it was possible to use the metrics that I just introduced to you to have an idea of about the quality of our segmentation results. Well, sometimes our object of interest don't have a well-known defined shape, geometrical shape. So some years ago, a very successful technique was proposed that is known as active contours, or also known as snakes or parametric models. So the idea is, okay, I will consider that my object of interest will be segmented by a contour. The contour is made of points. So I will define internal force that try to keep the points together in the contour. And then the contour will be located in the image, and then will deform towards the object of interest. Will be push and pull from the borders of the object of interest. And for that, I will have the internal force that try to maintain the points together, but I will have also external force that will be computed from the image data, again, like the force fields that I told you some minutes ago, that are calculated, that are established using mathematical operators. And using this force, I will have energy functional, so internal energy and external energy. I will be able to derive the motion equation that gives you the coordinates of the points along the segmentation process, using the formulation of Euler-Lagrange, and then I will have an iterative process. The idea is the contour will deform towards the borders of object of interest until you get an equilibrium of energy. That means that the points will stop to move, and then we get the segmentation done. That is here an idea. It's typical, the initialization is made by an expert, so you just define a root contour, and you can see that after the process run, you can get the final shape of your object of interest. Anyway, this approach works very well, is very successful, but there is a big disadvantage with this approach. It cannot handle, by default, it cannot handle variations of topology. That means if you start with a contour, typically a closed contour, at the end we'll also have a contour. But sometimes the idea is, okay, I start with a contour, but I want that the contours can be split because I want to segment other parts of the object of interest. And then a new approach was suggested that is known as the level set method or geometrical model, and is more or less the same. So I have also a functional of energy, I have internal and external energy, but now the difference is that to obtain the moving equation, I will integrate in the moving equation a level set function. And now the iterative process will stop when the level set function will get at 0. And with this topology, with this approach, I can handle variations of the topology. In these slides, you can see very clearly the difference between the traditional active contours and the level sets methods. This is a ultrasound image. The idea was to segment the borders of the carotid artery, and so the goal is to segment these borders here, okay, but also the borders of this internal region. You can believe me, we start with the same rectangle in both cases. Using the levels, the active contour, I was able to segment only these external borders because it starts with a rectangle, and at the end I just have one contour again. But now, starting with the same rectangle, you can see that using this level set methodology, I was able to obtain not only the external contours, but the initial contour was split, and then I will get also the internal borders that I have here. Here you can see an example using these deformable models to segment the lumen of the carotid artery. So this is a very difficult image to analyze because the resolution is very low. It's also ultrasound image. First of all, we apply a statistical method to obtain a first segmentation. Then we invert. You can see the possible regions, candidate regions to be the lumen, but we know that the lumen has more or less a circular shape. So using this information, we select this region as the possible region of interest. And to define the final segmentation, we start with the border of this region with the active contour to obtain the final contour of the segmentation. And then with the lumen identified, we were able, using again a level set method, to obtain the contour of the carotid. So using the same approach, we start with the lumen, and with the lumen we start a new level set to segment the contour of the carotid artery. 4 examples, and again you can see the result obtained by automatical method, but also by the expert. So, and you have here the values of some metrics, for example, the dice coefficient. Okay. Another approach to segment skin lesions. You can see a very different image, but the same problem. The idea was to segment these regions of interest. And you can see that besides the original image, we have here draw in red the contour obtained by our approach using a level set method. And here you can see the process going along the time. So it starts with a big circle. And then you see that the circle goes to the border of object of interest. And it is very, very robust, even with the presence of air, a lot of air, that as you can see here. Okay, but of course, this can be extended to 3D. Here we have 2 spheres for the lungs, to segment the lungs in CT image. Again, with the level set methodology, we are able to segment the organs. Another advantage of a level set methodology, besides the possibility to handle variations of the topology, is to, whenever you know some information of your object of interest, for example, if you know the common shape of your object, if you know the common position in the image of your object of interest, if you know some relations with other structures that are in, other objects that are in the input image that can be also segmented, you can use this information and integrate in your level set methodology and make the segmentation more robust. And here we have 3 examples, 3 MRI images of the pelvic cavity, and the idea was to segment these 3 organs at the same time, so the bladder, the vagina, and the rectum. And you see that the image even on the patient, we were able to segment the 3 organs. But we know that in this case, the relative position of the 3 organs is always this: bladder, vagina, and rectum. So we integrate this information in the level sets framework. And during the segmentation process, we always are seeing if the segmentation is going well and is not, it's according to this rule. And doing this, we can have a more robust segmentation. So yeah, and another example. So this is from the cardiac earth area, the cardiovascular system. We are segmenting at the same time the aortic tract, the right and left atriums. And as we know that between these three structures, we have always a thin wall. We are always doing the segmentation process using this level set methodology. We are always trying to avoiding the overlapping of the surfaces and we are always controlling the interaction between the three surfaces. And this is a iterative process, as you can see here, running. And at the same time, we have the three organs of interest segmented. And always according with the information that we know, that we have always a wall between the three structures. By the way, the final goal was not to segment the chambers, but to segment the wall that is between the chambers. But the wall is so thin that it's very difficult to segment. So instead, we segment the walls, and then with the wall segmented, we can get the wall. Of course, we also apply methodologies based on machine learning, support vector machine, optimal path for raster, and deep learning. Here we have an example of using a neural network to segment an image of metallographic image, so material science. The idea was to segment these two different classes, the background and this part. It is the element. I don't know. I think it's known as matrix or something like that. So using a neural network, it was a supervised neural network. If it is a supervised, that means that I will have a training data set. I will input, during the training phase, I will input to the network image that were segmented by an expert. So I classify the pixels of the image as belong to one of the classes or to the other. And then I will input to the image, to the network, the result that I know for each of the input image. So I need to have a train data set that was manually segmented. And during the training step, there is an algorithm of training that will try to find the best weights between the connections that I have in my network. So there are some empirical heuristic rules to define the topology of our neural network. And then during the training phase, I will establish the connection between the elements of the layers that are known as neurons. And the idea is to mimic what happens in our brain. Of course, it's a rude simulation, but it works very well in some applications. And when you have a new image that was not part of the training imaging, you input your neural network and you automatically obtain the segmentation, as you can see here. Of course, now with the computers that are becoming more and more powerful, you can have more networks, more layers and more layers. You can go to deeper layers. And with the big data phenomena, you have a lot of data that you can use to train your networks. So you can go to deep learning. And of course, my students are also using deep learning to segment the objects of interest. There are several topologies, architectures nowadays for deep learning. One of them that is very, very known is convolution neural network. So you have an input image and you apply convolutions operations and encoders and decoders operation, and you will obtain the segmentation that you want. Of course, you have also a training phase. You need also a training data set for this. And that's why it's important to have the big data, because if you want to obtain very good results, you need to have a very big training data set. Of course, there are some strategies for data augmentation to try to extend your training data. And then you have very complex networks that needs a lot of computer resource for the computation. And here is an example. So this is a CT image. The goal is to segment the liver. This is the manually segmented, and now here it was automatically segmented by our convolutional network. And you can see also in red the segmentation done by the experts, so you can compare the results. Here we are also using a convolutional network, but now to segment in MRI image the prostate. So here is the border of one of the image, and another one here, an example. Here we are using again a convolutional network, but now to segment in 3D, 5 different organs at the same time, spinal cord, both lungs, esophagus, and heart, and it works very well. Deep learning works. It's an approach that nowadays everyone is using, and if you have a very good trained data set, you can obtain very good results. Of course, generalization can be a problem, but it works very well. And here, another example. Now, we are not only using a convolutional neural network. The idea is to segment 3 objects of interest in the brain in MRI image. So for that, first of all, we perform a first segmentation using a convolutional neural network. But you can see that then we apply support vector machine for the final classification, and also she's boosts for the final classification. So you can have a problem, an application where you have several strategies combined to obtain the final segmentation. And another example that we are just doing now is not yet published. It is a different kind of application. It's related to security, safe cities. And we went from cameras of surveillance imaging device. You want to analyze crowds, detection crowds, and for that we need to, for example, to count the people that are in the image. And you can see we are developing a new deep learning network for this, and we can see that we are obtaining the first results are very promising. We can obtain the number of the people that are estimated to be in the image, and you can also build crowd density map. Here is another use of deep learning now for anonymization, because in this project we are using cameras, as I told you, for surveillance. We are obtaining all the time image. In some of the image we obtain personal data like faces, like license plates, and you need to remove this information because you need to protect the data, the personal data of the persons that can be imaged. And for that we are using a convolutional neural network also to detect the face and the license plates. And you can see the original image, and now you can see that we detect the license plate, and then we apply the blurring to not obtain the original data, and also for the faces. Okay, conclusions. So this area of image segmentation is very complex and demands. And as I hope you can see, realize from my presentation, all the algorithms are defined based on the application. So you cannot have a master segmentation algorithm. You cannot have an algorithm that obtains you very good results of segmentation for different kind of image. Of course, for the user, this is some kind of frustration because they cannot have only one algorithm. But for us, for the researchers, this is very, very interesting, because otherwise we cannot develop new algorithms. Of course, to obtain robust results, it's very important that get information, prior information about the object of interest, because you can integrate in segmentation algorithm and make the segmentation more robust. Of course, with deep learning is a very promising approach, but of course, to obtain very good results of using a deep learning network, you need to have very good and large image data sets for training. Okay, just the names of my students that have been working with me. Most of them are master and PhD students. Just a thank to my students, but also to the funding agencies that have been funding this research. Just the last words about some publications and events that we have organized. Okay, this is a journal that I started with Teles and Francis 8 years ago. It's devoted to imaging and visualization in biomedical areas. So if you want to segment, if you want to publish a paper related to image analysis in biomedical area, please feel free to submit. It will be welcome. And from the last year, January of last year, I started to be the editor-in-chief of this journal of computer medicine, biomechanics, and biomedical engineering. So it's not related with imaging, but if some of you is working on these fields using computer medicine in biomedical area, please, your submission will be welcome. And in 2012, we started a book series with Springer that tries to make joint efforts made on mechanical engineering and in medical image analysis. So it's known as Lecture Notes in Computational Vision and Biomechanics. This book series is open for proceedings, it's open for manuscripts, for books, for contributed volumes, and so on. So if you want to publish a book related to imaging or biomechanics, the book series is also open for submission. As I told you, besides the publications, we run some events related to biomechanics, biomedical engineering, imaging processing. This is a conference that will be held here in Porto in May. Well, because of COVID situation, will be online, unfortunately. But we also will have this conference at the end of the year. I think, I hope that we can join there physically, but I'm not so sure. In September, that is related with biomechanics and again with medical imaging analysis. And besides conference, we also run some events, thematic events in other big conferences. So I have some grants available for master and PhD holders. So if you want to work with computer in these fields of computer vision, is related to this is a project, joint project between Bosch company and my university. So I have, as I told you, some grants available. Please, if you are interested, just email me. And that's all. So thank you very much for your attention. I will say in Portuguese, obrigado. Obrigado for your attention. Thank you. This is, by the way, this is a picture of the old part of the town that is known as Ribeira. And this is the Douro River. And this is the typical boats that very years ago, some years ago, I don't know, 40 or 50 or 60 or more, were used to transport the port wine. So thank you very much for your attention. I'll be glad to answer some comments or questions. Thank you.
Thank you so much, sir, for explaining in brief about the computer vision and image segmentation approaches. And I'm pretty sure it will definitely help the research scholars working on computer vision or people who are to work in the field of image processing. So if anyone has any questions, you can take it now. Any participants, if you have any queries, you can take it. One. If we have no questions, anyway, sir has shared his email ID. Hopefully, I think you can contact him. So I believe he'll be happy to get you back. So before I conclude, I would like to request Dr. Tadarsi to kindly join us for the validated session of this international conference on next generation of IoT, which will be starting within 20 minutes from now, sir. So you can
sure.
take a break now.
It would be a pleasure.
Kindly do join us. Sir.
Okay.
Thank you very much.
Thank you very much.